first virus were the first virus are identified by Ivanovsky. Who is that? Ivanovsky, not I want a whiskey. Ivanovsky, the first virus separated, isolated by, who is that? Ivanovsky. You know the first virus name given it as Contagium vivum fluidum. Contagium vivum fluidum. First given name it as Contagium vivum fluidum. Okay. Contagium vivum fluidum. That Contagium vivum fluidum. Why they are given the name of Contagium vivum fluidum? Do you know? Why? Because it is one of the what happening? Why given the name? It is contaminating, it is causing disease through the fluid, whatever the fluid is there, that fluid, if it touch to the healthy plants, automatically healthy plants also being affected with this liquid material. The fluid or liquid which are not filterable by the bacterial filters, that are calling as Contagium vivum fluidum. What is that? The Contagium vivum fluidum. You know the contaminating the genetic material through the fluid touch from one to other. You know the virus are acellular organism. Acellular. There is no cellular structure, and these all are what we call as intracellular parasites. These are not, they can't live as a living organism at outside of the host. This can survive only in the host. Outside, this will be as like a crystals. No living organic mechanism, no synthesis material reactions will be happen. Only the crystal-like structure will be presented outside. And outside the organism, it will be look like a non-living organism, just like crystal, just like a stone. But whenever it enter into the host, it cause the diseases to the host. It totally depending upon the host for their metabolic reaction. There is no metabolic reactions at outside. Only if it is enter into the host, then only it start their synthesis of their own genetic material. There is no other cell mechanisms are present. Only the multiplication of the genetic material, especially in the hosts, only happen. You know, the viruses are non-cellular material particles which are made up of it, genetic material and protein that cause so many diseases to the living beings that enter into the living cells. You know, viruses are nothing but nucleoproteins, except nucleoproteins, there is no other things are present. Only protein coated and nucleus inside. That's it. No other cell structure, no other cell organelles, no other cell membrane, plasma membrane. These all are not there. Protein coat is acting as a coat, capsule, covering layer, and as like the covering sheath. And the nucleus, that means not the nucleus, nuclear material, not the nucleus. Nucleus is not there. Only nucleotides are present, either nucleic acids, either DNA or RNA only present. Either DNA or RNA. One thing you should remember, whereas in other organism, except the viruses, DNA and RNA both are present. DNA and RNA both are present. But in viruses, either DNA or RNA, only present, not the both. Either DNA or RNA only present. You know, the most of the plant viruses, RNA is present. The very important in so many exams they ask plant viruses. Most of the plant viruses are in the DNA, RNA, DNA and RNA are protein only. Which one? In the plant viruses, RNA is present, most of the plant viruses. But whereas in the cauliflower mosaic, wound tumor mosaic viruses, it having the DNA, even it is a plant virus. Whereas in the polio virus, influenza virus, poxo virus, these all are animal viruses, though it having the RNA as a genetic material. There is a variation is there. That type of the question will ask in the competitive exams also. Polio virus, poxo virus, or influenza virus genetic material is DNA or RNA or DNA and RNA. Which one? DNA, not DNA, RNA. Okay. Like that, there is a variations are there. Some little bit variations are there. Usually, plant virus having the RNA as a genetic material, animal virus having the DNA as a genetic material. Okay, these are I already told you very small, smaller than the small bacteria, very smaller than the small bacteria, non-living at outside. It enter inside and it reproduce inside the living cell, not at outside. It multiplies their genetic material inside the host. It multiplies, it takes replication, it will take place in the host only. It duplicate, it multiplies their genetic material inside. Okay. Within the short span of the time, single virus can produce thousands of the viruses. You see, discovery, the first virus term coined by, who is that? Beijering. Beijering, actually, he has given the name first it as, what it as? First it as Contangium vivum fluidum. What is that? Contangium vivum fluidum. First named it as Contangium vivum fluidum. Contangium vivum fluidum. Named it as Contangium vivum fluidum. Oh, sorry. Then in the next step it will come. No? Contangium vivum fluidum. Why he named it as a Contangium vivum fluidum? Do you know? He applied the juice which are affecting by the viruses that apply to the healthy plant. Even healthy plants also cause the disease. Whenever that fluid which are extracted from the viral diseased plant, then that healthy plant also affected with that fluid. That's why they named it as Contangium vivum fluidum. What it has? Contangium vivum fluidum. Even in the filter juice, which are filtered by the bacterial filters, which separate the bacteria from the fluids. From that filter is separated, though the viruses are not separated from that filter. That's why, these are very small, minute than the smallest bacteria. So that fluid, whatever the fluid that caused the disease, that affected the healthy plants. That's why they given the name it as Contangium vivum fluidum. Later, if the fluid it has a poison. The fluid it has a poison. Why? It causing the disease, it causing the sickness, it causing the death of the plant. That's why they given the name it as virus. Virus means in the Latin Greek, virus means poison. Virus means the poison. That's why named it as virus. Virus are interconnecting link between the living and non-living organism. Between the living and non-living organism, it having the both the characteristics. It having the both the characteristics. Living organism characteristics are there only if it is present inside the host. And non-living are acting as a non-living organism if it is outside the host. We can crystallize. You see the first Stanley crystallize the virus. Crystallize, make it as a crystal, stone-like structure, especially from the diseased tobacco plant. From the diseased tobacco plant, you see from the diseased tobacco plant, a crystallized the tobacco mosaic virus, TMV. What it is? TMV, tobacco mosaic virus. The tobacco mosaic virus is first crystallized virus. Tobacco mosaic virus is the first crystallized virus. Virus first crystallized by, who is that? Stanley. Virus first crystallized by Stanley. Stanley crystallized the virus from the infected tobacco plant. From the infected tobacco plant is separated and crystallized the disease causing virus. Okay. Then is said the virus are made up of nucleic acid and protein. Virus are made up of nucleic acid and protein. That means nucleoprotein. Nucleoprotein, which having the protein cover sheet and nucleic acid inside. Either DNA or RNA, nucleic acid inside, and which cover with the protein layer, which cover with the protein layer, only nucleoproteins, no other cell organelles are present, no cell wall, no cell membrane, no cell organelles, no nucleus. There is no cellular structure. That's why we call it acellular, non-cellular structure. The virus are not having the any cellular structure. Acellular, only it having the nucleoproteins. The first virus vaccine discovered by, who is that? First virus vaccine discovered by, who is that? The Jenner, Edward Jenner. Edward Jenner, he first discovered the virus, first developed the virus vaccine. The smallpox vaccine. అమ్మవారు అంటాం కదా, చిన్న అమ్మవారు పెద్ద అమ్మవారు, స్మాల్ పాక్స్, చికెన్ పాక్స్. You see, the vaccination, first vaccine identified, developed by the Edward Jenner. You see how the smallpox symptoms, smallpox symptoms upon skin, you see, and hands, small bubbles, sap bubbles, which are there, sap inside. There's a smallpox disease. the first vaccine developed for the smallpox virus by Edward Jenner. What he has done? He has done simple, whatever the deadly viruses, whatever the virus which was causing disease, that viral protein extracted from the infected virus, that we call it as interferons. What we call it as interferons. The interferon is a protein. Interferon is a protein. Interferon is a protein which released by the host, by the host at the time of infection. Whenever the host infected, that time, whenever the host is infected, that time the protein is released. That protein is called as interferon. Interferon proteins. The interferons extracted the protein and that injected to the already infected patients. So that what happened? The smallpox eradicated. That means cured from the smallpox. That means the cure from the smallpox. You see, the disease causing virus protein is curing the disease of the virus. You see, attenuated, dead virus is curing the same virus disease. You know, that's why, you know, our old parents, what they call, once you get the smallpox, whatever, chicken pox, the peddamavaru, chinnamavaru. Once you get the chinnamavaru, small bubbles upon your skin in the childhood, you can't get the smallpox in future lifetime. Why? Whenever you got infected with the smallpox, that time, what happened? The interferons are released in your body. That interferon will protect from that smallpox. That's the reason. Okay. And then you can see the so many historical aspects are there. Viruses, you know, the virus is very small, minute organism. This is much smaller than the bacteria, smallest bacteria. 1 million in an inch size. In the inch, 1 million in inch. Okay. These are 20 to 400 nanometers in size. You see, virus having the both the characteristics I told you. These are interlinks, interconnecting link between the living and non-living organism. These having the non-living characteristics and living characteristics. Both are there. Non-living characteristics and living characteristics. Both are there. You see, what are the non-living characteristics? What are the non-living characteristics of this virus? These are non-cellular. There is no cellular structure. It having the, what it have? What is it having? It having the capsid, protein coat, and nucleic acid inside. Nucleic acid inside. One more thing, either DNA or RNA, both are not present. Not the both. Any one, only present. Okay. And these are capable of reproducing only when inside the host. If it is not, if it is outside, it will not reproduce, it will not regenerate, if it will not produce their upswings. Only whenever it is in the host, then only it will regenerate their own viruses. Okay, you see, This is the virus structure. You can see DNA is present inside. DNA means this is animal viruses. Okay. If it is RNA, that is plant viruses. You see the capsid. Capsid means the protein coat. Covering sheath, which is made up of with protein, is capsid. And the envelopes also is present. These envelopes are present only in the animal viruses, not in the plant viruses. These are those special characters between the plant virus and animal virus. The animal virus which having the extra coat, that is envelope above the capsid. And some of the spikes, the coronavirus, this much spiky structure, as like a corona, kiritam. Okay, you see protective layer envelope is present in animal cells. Okay, and the most of the virus infect only the specific host cells. Not all the cells will be infected. Not all the organisms will be infected. Only the specific host cell. Suppose some of them cause respiratory, some of them cause skin, some of them cause other organelles. Okay, you see HIV virus, Ebola virus. What is the another non-living structure? Inactive outside the host. These are inactive. These lack the ribosomes, enzymes which are needed for the metabolism. So there is no metabolism at outside. There is no metabolism. But it can reproduce inside the host and which use the enzyme of the host for their reproduction, for their multiplication of their own offspring. It utilizes the host enzymes, host raw material, host enzyme and host raw material. Okay. So many diseases which caused by the viruses, smallpox, measles, mumps, chikungunya, influenza, cold, cough, AIDS, Ebola, flu, measles, polio, hepatitis, and some of them cause cancer. See, viruses are different from other things in nature. The various shapes are there that are rod shape, round shape, tadpole shape, and many-sided, multifolded, and helical, and cuboid, cube-like structure, so on and so forth. So many helical shapes are, you see, helical shape in Ebola, polyhedral shape in influenza, complex shape in bacteriophage. You see, helical viruses, the tobacco mosaic virus. You see, Ebola virus, polyhedral virus, multiple angle shape, polyhedral. You see, the tadpole shape, bacteriophages, and you can see, uh, just a wait. I will show you the other. Okay. Could you able to see this?
No, sir.
No, it's not appearing.
Not
Okay,
appearing, sir.
okay. Just again I will share where it is. Okay, now it is okay. Okay, now you can see the differences. These all are the topics we having: origin of virus, introduction of virus, history of viruses, virus classification, alter structure of the virus, chemical composition of the virus, isolation and purification of virus, replication of virus, transmission of virus, and general account of plant and animal, human viral diseases. These all are the topics. Of that first introduction we have studied. What are the viruses that we have studied? These are simple and acellular, no cellular structure. Okay, these are infection agents. It infects the living cells. Okay, and these are log all the enzymes and ribosomes, cell organelles inside, but it modify the host enzymes into their own purposes, for their own purposes. They utilize the enzyme, host enzymes for their reproduction, replication purposes, multiplication purposes. I already told you there is no cell wall, there is no cell envelope, there is no specific cell structure. Okay, that's why acellular. You see, so many theories are there on the origin of viruses. That is a still now the virus origin is conflict, unclear. But so many theories are there. That is regressive hypothesis. That means regressive means degeneracy, reduction. Sometimes they may be small cellular organisms, maybe degenerate and reduce their structure, and it will become the viruses. And there is a supporting evidences that mean that these are smallest organism than the other, especially in the rickettsia bacteria and chlamydia and rickettsia actinomyces like bacteria that all are parasitic bacteria that all are reproduced only inside rickettsia and chlamydia bacteria that are reproduced inside only. It depending upon the host only for their reproduction. Like that, the virus also having the showing the same. And the next is escaped gene theory. Escaped gene theory means here the genetic material is escaping, genetic material is missing. Here, what happened? Either DNA or RNA. From the larger organism, either DNA or RNA escape. Either DNA or RNA escape. That time, what happened here? Use toolbar. Close toolbar. Okay. And that what happened? The whatever the largest genetic material is degrading, and some of the genetic material is damaged, some of the genetic material is misplaced, so that either DNA or RNA will be remain of the genetic material. It has not having the much larger genetic material. And the next is third one is co-evolution hypothesis. Co-evolution means these are evolved along with other bacteria and other cellular organisms. Okay, though it are cellular, these are also evolved along with the cellular organelles. You see, the archaebacteria and eukaryotes structures, replication mechanism, same as like. You see, I already told you the study about the virus is called as virology. Okay, virus word derived from the Latin word virus means the poison. The first virus name given it as a contagium vivum fluidum by who is that? Bygerin. You know the recently COVID-19, the virus that caused much damage, that caused to death of the so many people in so many countries. Okay, you see the Bygerin. I already told you the Bygerin. Bygerin We call that fluid which causing the disease as what it is. As what it is. Contagium vivum fluidum. Soluble living organism, which is soluble, which is causing the disease, it is contaminating, it is spreading from one to other. That contagium vivum fluidum first given the name to the virus. Okay, first identified the infection causing by the viruses by Bigering. What is that? Bigering. And the later it named it as virus. Okay, the first virus structure observed on the electron microscopy by, you see, Nolan Rusk. You know who is the Nolan Rusk? First electron microscopy discovered by Nolan Rusk. You know, Nolan Rusk. And then after that, the Stanley. The Stanley crystallized the tobacco mosaic virus. Crystallized the tobacco mosaic virus. Okay, filter with the bacterial filter, though it having the fluid which causing the disease, to plants and animals. Plant, you see, who are those? Who is that? Are you there? Frederick identified the virus which are causing disease, which are infecting the bacteria, which are infecting the bacteria. That means it depending upon the bacteria. That's why the given the name it as bacteriophages. Bacteriophages means not the bacteria. It's living upon bacteria. Phages which are depending upon the bacteria. That are bacteriophage. That are what we call it as the bacteriophages. Okay, so a lot of history is there about the virus. Lot of damage also happen by the viruses. You see, which are so many 2,000 above varieties of the viruses which are causing disease to the animal, plant, and human being are identified. Okay, these all the characteristics I already told you. These are acellular, non-cytoplasmic infection agent. There is no cytoplasmic cell organelles. Okay, virus is nothing but nucleoprotein. Virus is nothing but nucleoprotein. Okay, these all are very smaller than the smallest bacteria. These all I already told you. No? Virus may be crystallized, first crystallized by Stanley. Okay, and virus cannot contain the both the nucleic acid, either DNA or RNA present. I already told you in the plant virus, RNA. In the animal viruses, DNA is present. But some of the variation is there in some of the plant virus. DNA is there, cauliflower mosaic and wound tumor viruses. And then, whereas animal virus also having the RNA instead of DNA. You know, all the viruses are obligate parasites, intracellular obligate parasites. There is no symbiont, there is no autotroph and other heterotroph. It totally depending upon the host. If it is present, the host is present, then only it will be survive, it will be multiply. Only within the host cell. That's why viruses are obligate intracellular obligate parasite. Now outside it will be like a crystals. There is no metabolic reactions at outside. It having the both the living and non-living characters. That's why I already told you these are the connecting links between the living and non-living organism. What are those characteristics that we will see? What are the characteristics of the living and non-living we already shown, we already seen, we already studied. But again, we will see. Okay, what are those characteristics? The living and non-living organism. You see, what are those? What are the living characteristics? It having the genetic material as a DNA or RNA. Okay, and they can undergo mutations, and they show irritability, causes itches, whatever it is, causes damage, causes infections. They are capable of reproduce. Where it can reproduce? At outside? No, inside the host. It shows the irritability. It undergoes the mutation. It having the genetic material. It having the genetic material, not the both, only one. And they can transmit it from one host to other. They can transmit from one host to other by any mechanism that we will discuss. Transmission, that we will discuss transmission of viruses. Transmission by physical or by living a vector or non-vector. Vector-based transmission or non-vector-based transmission. That also is a question. By air or by water or by any insects or aphids or arthropods or mosquitoes, parasitic plant, or by Japanima, Langdora, nematodes. So many carriers, transmission factors are there to transmit. And you can see they react to heat, chemical, and radiation. With the heat or with the chemical or with the radiation, we can control the virus. See, these develop resistant to antibiotics. These are developed resistant to antibiotics. These all are the living characteristics. These all are the characteristics of living beings. Along with that, is also having the non-living characteristics. Along with that, also having the non-living characteristics. What are those? We can crystallize as like a stone. They are inert outside the host. Can't do any metabolic reaction at outside. They lack cell membrane, no cell membrane, no cell organelle, no cell wall, no cell structure. Total cell structure is absent. So that's why no cytoplasm, no organelles. They don't show any cell division. They can't grow in size and shape at outside. They don't possess any sort of nutrients. No need of the nutrients to them. It utilizes the enzymes and proteins of the host, and it multiplies their own genetic material. You see, don't respire and exit. No metabolic reaction other than the replication. It just replicate. You see, don't undergo their own metabolism. It depends on host metabolic reaction. It absorbs the protein and enzyme only from the host metabolism. Any energy producing systems are not there. No energy producing systems inside the viruses. No energy producing system. It utilizes the energy of the host. It can't synthesize any energy. It can't do multiply. It can't do divisions at outside. There is no any reactions, respiration, nutrition, no need of the nutrients, no cytoplasm, cell organelle, cell wall, plasma membrane. Okay, no activities at outside. If it is there in outside air, no activities will be done. It will be remain as like a stone, as like a non-living being. But whenever it enter inside the host, it will perform all the actions. These are the characteristic features of this virus. It having the, what are those? What it having? Living characteristics and non-living characteristics. Both are there. Okay, that's why we're calling it as interconnecting link between the living and non-living organism. Interconnecting link between the living and non-living organism. Okay, then you can see the classification of virus. Classification based upon so many, uh, either based upon the host or based upon the structure or based upon the specificity, genetic material, nucleic acid material. So many types are there. Virus classification, you see, based upon the host. If you see, if it is causing the diseases based upon the pathological symptoms, epidemiological symptoms, virus, if it is causing the disease to the plant, that we calling it as plant viruses. Phytophage, phytophages. You see, if it is there on invertebrates, that means you know, from the protozoa to echinodermata, and vertebrate from the fishes to animals, and dual host. It will infect the two different hosts, and plant and animal are different type of the animals, insect and birds are like that. Okay, and then you can see the Holmes classification. What he said, phagine. That means it infecting any host. If it is infecting the bacteria, that is bacteriophage. If it is infecting the plant, phytophage. If it is infecting the animal, zoophage. If it is infecting the cyanobacteria, cyanophage. And like that, phage, phages. That means infection, based upon the infection. Phytophage, zoophage, cyanophage, bacteriophage. And then the modern classification, ICTV classification, that means International Committee on Taxonomy of Viruses, that taken consideration of their structure based upon the existence and based upon their structure, they have classified from the phylum to the species levels, and then based upon the International Code of Nomenclature, ICN and ICJN and Microbiological Society of India, viral classification is done with the consideration of Microbiology Association, the Zoological Association, and Botanical Association. You see, what are those? The 2019 viral taxonomy, they separated into how many? 24 families. Of the 24 families, you see what are those, from alpha pseudacea to transmitted day. There you see there is a various type of the class based upon the structure. You see various type of the structures are there: rod shape, helical shape, icosahedral shape, circular shape. So many types structures are there. Cuboid structures are there. Mixed structures, tadpole style-like structures are there. And then what the majority of the viral classification we are following is Baltimore classification. Baltimore classification system classified into 7 groups based upon their nucleic acid, material, either DNA or RNA, based upon nucleic acid material, either DNA or RNA. Based upon that, they classified, categorized the viruses into 7 groups What are those 7 groups? First one is double stranded DNA viruses. DNA double stranded DNA viruses. That is adenovirus, herpes virus. You see, and single stranded DNA virus, parvoviruses. Parvoviruses. You know, the largest virus is the parvovirus. You see, uh, smallest virus is the polio virus. You know, the polio virus. And then RNA as a genetic material. That means usually plant viruses. You see, double-stranded RNA. Usually RNA is a single-stranded, but double-stranded RNA, reoviruses, and single-stranded RNA. You see, in the single-stranded, again, positive sense single-stranded RNA and negative sense single-stranded RNA. You see, positive sense single-stranded RNA, coronavirus, COVID-19, and pecan viruses. You see, the negative sense viruses are orthomyxoviruses and rob dog viruses. You see, and reverse transcripting diplo single stranded RNA. You know the reverse transcription? Temin Baltimore first identified in the viruses. You know what does it mean, the transcription? Do you know the transcription means? You know the protein synthesis? Protein synthesis.
DNA to RNA transcription.
Yeah, DNA to RNA is the transcription. And RNA to amino acid protein synthesis is translation. These both reactions commonly called as protein synthesis, central dogma of protein. Protein synthesis are also called as central dogma of proteins. That reaction is very, very important in the molecular biology, genetics, protein synthesis, translation and transcription. Okay, this transcription usually from DNA to RNA. That is transcription. Okay, DNA to RNA. Okay, otherwise we type DNA to RNA usually. DNA to RNA. That is transcription. If it is up and reverse from RNA to DNA, from RNA to DNA, reverse transcription. That reverse transcription first identified in the virus by Temin and Baltimore. Temin and Baltimore. It's very, very important. These two scientists identified the reverse transcription reaction in the virus. That's why virus infecting the living beings, they're reversing their genetic material RNA will become the DNA material, so that whatever the host DNA is getting damaged, with that it is totally damaging, causing the death of the host by degenerating the host DNA. That is the most tragic reaction, most effectful reaction for the living organism, that is reverse transcription. Totally degrading the genetic material. Their own RNA material is converting into DNA. That means it's converting their own genetic material. It's spoiling the host genetic material. That is the reverse transcription reaction that happen in the viruses. That's why virus are calling it as poison. Okay, and then reverse transcripting circular double-stranded DNA and single-stranded RNA. These are the types. These are the various types of the viruses as per Baltimore classification. As per Baltimore classification. Okay. And then we have to study more. A lot of syllabus is there, no? More one class only there. You see, the DNA virus, double-stranded and single-stranded DNA. You see the double-stranded Again, if it having the envelope, protein envelope, that are enveloped viruses, poxovirus. You can see unenveloped. There is no envelope outside the nuclear protein capsid. You see, papillomavirus, adenovirus, and unenveloped is parvovirus. There is no envelope outside the capsid. You see the RNA viruses. What are those? Plus stain and minus. Positive stain and negative stain RNAs. Okay. And both positive and negative. And both RNA and DNA types are there. Okay. And then you can see LHT system of classification. Here you can see based upon their characteristic nucleoprotein structure. Okay, deoxyvira, ribovira, DNA viruses and RNA viruses. Okay, based upon the, you see, again, symmetry. You know, symmetry also very important question. Symmetry of the viruses. It's having the three symmetries. Helical symmetry, icosahedral symmetry, and both the symmetries. You see various properties based upon their various properties. You would having, you see, helical symmetry and icosahedral symmetry, and you would having the both the symmetry complex. Symmetry means it can split into two equal. Now, if it is possible, that is symmetry. How? What say?